Hi, this is Dr. Ram here. So, I'm going to teach you about some important metabolism topics in biochemistry. Okay, so here is a cell. So, we have trillions of cells in the human body, and this pump, that is the sodium potassium pump, is very important in all excitable cells in the human body. And this pump needs energy because this pump is going to maintain high sodium outside and high potassium inside. And if high sodium is not maintained outside, then the cell swells up and damages because high sodium inside, followed by water, cell swells up and damages. So this pump needs energy. So we eat every day, like glucose, lipids, proteins. We eat every day to supply energy for this pump so that your nerves, heart, all excitable tissues will function and you live. And now, this glucose, lipids, and proteins are digested and then absorbed, digested and then absorbed in the gut, digested and absorbed in the gut, and then it enters the cells. After absorption, it enters into the cells and metabolized to form ATP, and this ATP is used to run this pump. Okay, this is the overall idea you should have. So for the digestion process, we have a lot of enzymes. For the carbohydrate digestion, we have amylase in the salivary gland and pancreas. For the lipid digestion, the stomach, the emulsification process that is happening in the stomach, and then the bile acids, the bile acid that is coming out from the gallbladder, and it surrounds the fatty acids, I mean the lipid droplets, and it's absorbed. And the protein you call as trypsin in the pancreas, and also the acids in the stomach is very important for the digestion of the proteins, and trypsin is also important for the digestion and absorption of proteins. So likewise, these carbohydrates, lipids, and proteins, all the smaller particles of carbohydrates, lipids, or proteins are absorbed in the gut and enters the cells. Fine. Once the glucose enters the cells, or the fatty acids, or the proteins, these can enter the cells and they get converted to energy. Okay. But before that, I want to separate the timings, I mean, in relation to the food. For example, when you have some food in the morning or the lunch, I mean, the insulin is a hormone that is going to be in action like 1.5 hours, or say 90 minutes around that food. Okay, after you take food, your insulin is released and you will be in insulin world for about 90 minutes after that. Okay. And this insulin is an anabolic hormone. This insulin is an anabolic hormone. So this is going to move the glucose into the cells. So here is a cell, here is a blood, and here is a glucose. And this glucose enters into the cell because of insulin action on its receptor. Insulin acts on its receptor and glucose enters into the cells and gets metabolized to energy. But note that not all the cells need insulin for the glucose to enter. Not all the cells. An important cell is a muscle cell which absolutely need insulin for the glucose to enter. Fine. And some of cells like RBCs, they don't need insulin for the glucose to enter. This is one of the main function of insulin, that to make glucose entry into the cells. Okay. And remember, how is this insulin released? So here is a glucose that's going to act on the pancreas. Some islets are there, some cells, group of cells which you call it as islets. In these islets you have alpha cells, beta cells, delta cells, And this islets helps to secrete a lot of hormones, like alpha cells releases glucagon, and beta cell secretes insulin, and delta cell secretes somatostatin. Fine. And this glucose is going to stimulate this beta cells to secrete insulin. Okay, and then the insulin comes into action and makes this glucose to enter into the cells, and glucose metabolized to energy. And now the carbohydrates. What are the carbohydrates? Glucose, galactose, and fructose. These are the monosaccharides. Okay, so these are the smallest particles that they are the carbohydrates are finally broken down to these monosaccharides in the gut. Okay, and these are get absorbed. Fine, and these can enter the cells. Other bigger polysaccharides cannot enter the cells, and these can enter the cells. Okay, now this glucose, galactose, fructose enters into the cells and gets converted to pyruvate. Okay, so this is happening in the cytoplasm. And this pyruvate can enter into the mitochondria. So this is a mitochondria. This pyruvate can enter, and this runs in the TCA cycle first, and the electrons or electrons carriers like NADH, like NADH or FADH2 are produced in the TCA cycle, and these run down in the ETC, the electron transport chain, so that ATP is created. And this ATP is used for so many processes, especially to run the sodium potassium pump. Is one of the main. And there's still so many thousands of process where energy is needed. Fine. So now the insulin is a very important hormone. Fine, that is going to make the entry glucose, galactose, fructose. It's going to make it's promote the entry of glucose. Okay, and this pyruvate is like running down in the TCAT cycle ATP. And remember, the insulin is a very important hormone for this process. And also remember, this insulin is an anabolic hormone. Whenever there is high energy in the cells, okay, whenever there is high energy in the cells, this insulin is going to convert the glucose to glycogen, okay, or to fatty acids or proteins. Fine. So the insulin is an anabolic hormone. It's going to store the glucose in the form of glycogen, or it can convert the glucose to fatty acids or the proteins. So this is an anabolic hormone. So this can be used in the future. For example, when you are not eating food, the fasting state, these can again be broken down, glycogen to glucose or the fatty acids to glucose, okay, or the fatty acids to energy or ketone bodies, okay, likewise, or proteins to glucose. And these can be used to produce the glucose, and glucose runs down in the TCA cycle, ETC cycle to create energy. But that's promoted by the fasting world. It's the main hormone is glucagon. Fine. Still other hormones like the growth hormone or the cortisol, the epinephrine also there. We can discuss it later. So this is about the carbohydrate metabolism. And you have a lot of enzymes like glucose. This is converted to pyruvate. Some of the important enzymes like the pyruvate kinase. Okay, it's 1 second. Okay, so glucokinase or hexokinase, it's a very important enzyme. Kinase means it's going to phosphorylate the glucose. Okay, so glucose is phosphorylated to glucose 6 phosphate, and when the glucose is phosphorylated, this is trapped inside the cells. Fine. And further, there are two important enzymes like PFK1 or PFK2, and one more important enzyme is pyruvate kinase. And these three important enzymes are going to convert the glucose to pyruvate. We'll talk about this in detail later. But remember, once a pyruvate is produced, it's moving into the mitochondria. And this pyruvate is converted to acetyl CoA by a very important enzyme, pyruvate dehydrogenase. Fine. And this pyruvate dehydrogenase is a very important enzyme, and it needs at least five cofactors for the functioning of this enzyme. It needs at least five cofactors. One is B1, B2, B3, B5, and lipoic acids. So thiamine, biotin, thiamine, riboflavin, niacin, okay, pantothenic acids, lipoic acids, and these are the cofactors that is needed by the PDH to convert the pyruvate to acetyl CoA. Any deficiency here, for example, PDH deficiency or these cofactors deficiency, okay, the pyruvate will not be converted to acetyl CoA. And this acetyl CoA is going to run in the TCA cycle, okay, to create to create NADH where it carries electrons, and FADH2 where it carries electrons. Fine. And now these electrons, NADH and FADH2, runs in the T cycle. Okay, this is mitochondria, this is inner membrane, this is outer membrane. Here are some complexes 1, 2, 3, 4, and 5. Fine. And these NADH and FADH2 provide electrons to complex 1 and 2. Fine. And then from one complex to another, these electrons travel with the help of some enzymes. Okay, from 1 to 2, 2 to 3, 3 to 4. When it travels, each time the hydrogen ions are pushed out. Hydrogen ions are pushed out. Fine. The hydrogen ions are pushed out. And this is the matrix, right? So these hydrogen ions have, why it's pushed out? Because to maintain the electrical gradients, okay, so hydrogen ions are pushed out. And in a complex phi, these hydrogen ions move in. Fine. These hydrogen ions moving in where this complex contains enzyme which is called ATP synthase. Fine. This ATP synthase is going to make ATPs. Fine. And remember, this is an ETC or electron transport chain is also called oxidative phosphorylation. Oxidative phosphorylation. So 1, 2, 3, and 4. This up to 4 here, this process 1, 2, 3, 4 is oxidation process. So this is 1, 2, 3, 4, and 5. And up to here is called oxidation process. Why? Because in complex 4, the oxygen is accepted so that water is produced. And oxygen is accepted. Okay. And remember, in complex 5, there is ATP synthesis. This is a phosphorylation process because ADP is here. Once the hydrogen ions enters, this produces the phosphate and the ATP is produced. Fine. So hydrogen ions enters in this position. So oxidative phosphorylation. So we have some drugs like dinitrophenol. Fine. Dinitrophenol. This is a very dangerous drug. Illegal use of this drug results in weight loss. Why? Because it's going to separate this 1, 2, 3, 4, and 5 at here. Okay, it's going to damage the mitochondrial membrane, going to separate this oxidation and phosphorylation so that heat is continuously generated and ATP is not generated. Fine. The same, the aspirin drug. Okay. And there is one important protein which is called thermogenin. This thermogenin is going to this is present in the brown fat. Fine. This thermogenin is present in brown fat, and it's going to make some pores here. It's going to make some pores here. Okay, so that oxygen is accepted and heat is generated at this step. A lot of heat is generated because of the continuous running of oxidation process. Okay, and this complex phi, there is no, there is a very low AT production because of this thermogenin is present in brown fat, and this brown fat will be seen in the newborns, okay, or in some animals which live in cold conditions. Fine. And one more important point, we have so many drugs and some proteins that is going to inhibit this ETC cycle. And one more important point is that carbon monoxide or this cyanide, this can inhibit the complex 4. So carbon monoxide or cyanide is really dangerous, and this can inhibit the complex 4. You remember that is also important. Okay, and dinitrophenol, thermogenin, aspirin is also very important. These inhibit complex 5 and these inhibits complex 4. So this is about the ETC cycle. Okay, so now you learned about the carbohydrate metabolism. Fine. And next section, learn about the lipid and protein metabolism. We are talking about the insulin world. Okay. And also remember this excess, the carbohydrate, I mean, the glucose, you produce energy when there is excess ATP, that is insulin is used to make the glucose to glycogen, or the fatty acids or the proteins. Okay, so we'll learn all about this in next session. And one more point I want to make. Okay, so I told you about the glucokinase and hexokinase. Fine, this is very important enzyme. This is going to add phosphates to the glucose. So glucose is converted to glucose 6 phosphate in the first step of glycolysis, and it's very important enzyme. And remember, this is present in um all cells, all cells of human body except liver and beta cells of pancreas. Okay, liver, important is liver here. Okay, and this is present in liver and beta cells of pancreas. I'm considering liver here. Fine. And this hexokinase is something like something, remember, like it's a very having very high affinity for the glucose. It's having very high affinity for the glucose. Fine. And this glucokinase is having low affinity for glucose. I mean, this hexokinase loves glucose more than the glucanase. It's absolutely love the glucose. Okay, for example, if this is the enzyme and here is a substrate, okay, and this substrate is converted to a product, fine, with the help of this enzyme. And now, if I say this enzyme has more affinity for the substrate, that means this substrate is quickly converted to a product because of a more affinity. So if affinity increases, the substrate concentration decreases, or I say the Km decreases. Fine. Let's talk about the Km or Vmax in detail in pharmacology session. But remember, substrate concentration decreases. The Km, that's a constant. Substrate concentration is a constant. Okay. Let's talk about later. And this decreases. So when hexokinase, so if I say hexokinase, which I want to comment about Km, it's decreased. And glucokinase has very low affinity, so the Km is increased. I mean, the substrate concentration increases. And when you talk about the Vmax, always it's a character of enzyme. Also remember, this Vmax is very high. I mean, the velocity for the velocity at which substrate is converted to a product is very high for the glucokinase and very low for the hexokinase. It's just its character of enzyme. Fine. So this is present in the liver, and this is present in all the cells except liver and beta cells of pancreas. Also, this is present in the beta cells of pancreas. Fine. So this is a very important point to note down. And also remember, this hormone can be stimulated by insulin. Okay. So glucokinase can be stimulated by the insulin, and hexokinase don't depend on the insulin. Fine. And this is one important point to make. Okay. Then about the important PFK1 and PFK2. Okay, this is one important enzyme where it's going to phosphorylate things. Okay, phosphorylate substrates. And remember, this PFK1 is present in um, all the cells. Okay, all the cells of human body, but this PFK2 is present in the liver. What is the function of this PFK2? Remember, this PFK2 is going to stimulate the PFK1. I'll tell you in detail. For example, this is glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 bisphosphate. Likewise, it goes in the glycolysis pathway. Okay, and this reaction, you have PFK1 enzymes, very important rate limiting enzyme. This is going to convert the fructose 6 phosphate to fructose 1 6 bisphosphate. Kinase means just adding a phosphate. Going to add one phosphate, so we call this bisphosphate in position 1. Fine. And this PFK1, when the cells is having very high energy, for example, high ATP or high citrate, fine, and it's having very high energy, this is going to stop. I mean, it's going to slow down the production of this fructose 1,6 bisphosphate. I mean, this step is inhibited. Fine. If this step is inhibited, do you think the glucose can be converted to glycogen or the fatty acid or the proteins? Because the liver needs glucose to produce all this. So even if there is high energy, I mean, increase ATP or citrate, this glycolysis process should be continuously running so that the glucose can be made to produce the glycogen, fatty acids, or proteins. Am I right? So this fructose 6 phosphate will be converted to fructose 2,6 bisphosphate by this enzyme phosphofructokinase 2. Okay, and this 2,6 bisphosphate is going to stimulate this phosphofructokinase 1. Do you get it? So there's no way this glycolysis should be stopped because glucose should be converting to glycogen or the fatty acids or proteins. So this process should be continuously running. So even if PFK1 stops the conversion of fructose 6 phosphate to 1,6 bisphosphate, this PFK2 produces this substrate, and this substrate is very important, very important, because it's a very important stimulator for this PFK1. Okay, so this point is super duper important, and this is present in the liver. Okay, one more important point about the pyruvate kinase. Okay, so glucose converted to pyruvate. Okay, pyruvate kinase. Okay, and this pyruvate kinase is a very important enzyme, especially in the RBCs. Fine. Actually, pyruvate kinase is present in all cells, but this pyruvate kinase is especially important in RBC because this RBC there is no mitochondria, and there is no, I mean, there is a lot of generation of ATPs in the mitochondria, right? But if there is no mitochondria, where is the energy coming from in the RBCs? Where is energy coming from? So the energy is coming from the substrate level phosphorylation of glycolysis. A very important point. Substrate. So this glycolysis pathway produces a lot of substrates, right? So those substrates can be used to produce this ATP, which is called substrate level phosphorylation. So if a pyruvate kinase is genetically absent, the RBC ATP is low. Okay, so when ATP is very low, you have sodium potassium pump here. So this actually pushes the sodium out, potassium in. So if this pump slows down, okay, the sodium gets accumulated inside the cells. So here is RBC. When the sodium is not moving out, sodium gets accumulated, so it's attracted by water. I mean, water follows to maintain the osmolar gradient. Okay, and the cell swells up, swells and damages. Okay, so when it is damaged, when it is damaged, okay, it causes hemolysis. Fine. And these are some important concepts you want to learn in your step 1 exam. Fine. I'll teach you some other things later in next class. Thank you.